ProHits. Prokaryote proteomics at high temperature for single cells. Can you briefly introduce yourself?
Hello, my name is Josephine. I'm here at the Vrije Universiteit Brussels in Belgium, where I'm currently stationed within the ProHits framework, and I'm DC number 2. Before that, I have spent a brief year as a research assistant investigating thermophiles, and I trained as a molecular biologist.
Could you introduce your project and explain your role within the ProHits framework?
So as DC2, my main responsibility is to ensure proper cultivation of all the thermophiles that we investigate within ProHits. So I'm cultivating them all, and then I'm sampling them, and I'm distributing them to everybody who needs it in the mass spec framework. To the people who do mass spec acquisition, I also provide a lot of biological insight and have to research them, and also potentially find different thermophiles that we could investigate within ProHits.
How does your project connect with the work of other DCs?
My biggest connection within ProHits, I think, must be DC8, Anna, who is in Vienna. So she's the other biologist of the group, and she mostly focuses on the archaea. But she and I, we both do the sampling of all the thermophiles of ProHits that will be analyzed and investigated. I'm also in close contact with DC6 Dino, who's in Strasbourg, and also DC10 Mansi, who is in Debrecen, as they both receive my samples and do the sample prep for the mass spec acquisition. However, I'm also in contact with DC9 Francis to help him cultivate our different thermophiles for his single cell experiments. And then I'm also in contact with the computational people, in which I will, maybe not so much now, but in the future I will be providing some, or not just providing, of course it's a collaboration, so I'll be talking with them and looking at their data analysis, and together we'll figure out what is happening when our thermophiles meet.
What secondments or trainings do you have planned, and what are your expectations from them?
My first secondment has actually already taken place, and that was with DC6 Dino, where I went to Strasbourg for a month and shadowed him in the lab and learned how to do the sample prepping for mass spec injection, understanding how mass spectrometry works in more detail, which has been truly fascinating and a really great learning experience for me, because that complements me both doing the sample prepping but also understanding what needs to be done when the injection has to occur, and then also see how you analyze the output and how you name all the proteins that you've detected in your samples. I will also, further along in the project, I will be joining DC8 Anna in Vienna. And Anna is of course working at NovoArc, where they're looking more at the biotechnological usage of, well, specifically archaea. But that's going to be really interesting to see how you go from fundamental biology, as I mostly work with here at VUB, and how you implement that in a biotech startup.
What motivated you to join this project, and what are you most looking forward to during and after your PhD?
Well, as a biologist, I have always been fascinated by all aspects of life, whether it's us, but also the extreme parts. And in complementation of that, I of course also strive to figure out a way to protect and preserve our biodiversity on this planet, which is what I actually truly find exhilarating about ProHits, because it combines both. I both have an opportunity to dive into a type of organism that lives in just such extreme parts of this globe that you wouldn't imagine that there could be life there, whilst also try and figure out how we can utilize those into green biotechnology that hopefully can become a production pathway that will decrease carbon emissions, which will be part of the whole climate change mitigation strategy that is so absolutely necessary for the future of the globe. After I finished my PhD here at VUB and in connection with ProHits, well, the plan so far is to continue in academia with a postdoc. I hope to take one of the thermophiles that we're currently experimenting with here in ProHits and continue that and really, let's say, milk the potential of using that as a bioproduction chassis.
How do you plan to ensure that your project achieves its expected outcomes?
So in a consortium as big as ours, I think communication is incredibly important. So to be ensured that the project achieves its expected outcomes, I will definitely make sure that I will be as available as possible. Since I am the factory of the whole project, I will of course always be available to whatever type of sample that might be needed. But in terms, I will also of course be investing a lot of time in hearing what everybody has produced so far and figure out what kind of biological samples we then next want to produce, so we can further the depth of the investigation in all of our thermophiles. We'll of course continue reading to keep up with relevant literature.